Hi, I would just like to thank the organisers for this opportunity to present my work again, having really enjoyed the experience last year. Today I'm going to be talking about the field of biocodicology and how it can help or add another layer of interpretation to the history of manuscripts. The first thing to define is what is biocodicology. Well, we like to define it as the study of biomolecular information that forms part of the biological history of a manuscript. This can encompass numerous different techniques and analyses, but in this presentation, I'm going to focus on two of the most important ones that we routinely use. Now, of those two aspects we will be exploring here, one is genetic analysis and the second is proteomic analysis. The obvious first choice when people think of biological analyses is genetic testing. DNA is something that most people have heard of and have an understanding of. So often when we're approached to carry out a biocodicological analyses, the first question that tends to crop up is, can you do the DNA? Now, DNA analysis is definitely an important contributor and can give you a lot of information about species ID, the sex of the animal used to make the parchment, and in some cases even the breed identification of an animal. That part involves what we call endogenous DNA. So this is the DNA that belongs to the animal itself that's been used to make the parchment. And that is the kind of information we can obtain from there. But in addition to that, there's also another part of the DNA that's lurking on the surface of these documents. So some of that is obviously human DNA, because over time, obviously from its construction, from its writing, and then throughout its history, people have been touching and using these manuscripts and will often leave traces of their DNA. So a lot of that still lies dormant on these manuscripts. However, human DNA is not something we tend to look at. It's not something we specialize in. And we have to make clear that the human DNA that would be on these manuscripts is a mixture of everybody who has ever come into contact with. So the importance is limited to the fact that you cannot readily separate out these individuals. But in addition to this human DNA, there's also another type of exogenous DNA, and that is the microbiome. And this is something we have looked at on occasion. This is the collection of microorganisms that live in and on the manuscripts. And these are often important because they can give us clues as to how the book has been stored or what kind of environment this book has been, or this manuscript has been present in throughout its history. So the microbiome is actually quite an interesting point to analyse. Now, the second type of analysis, which I'll be discussing more in depth, is the protein analysis, and that is the field that I predominantly work in. Now, by looking at collagen, which is the majority protein of skin, and at the end of the day, parchment is just animal skin, we can easily obtain this protein because there's so much of it, and we can use it to determine the species of animal used to make that parchment. In addition to the species, we can also get information relating to how that parchment was actually produced. So relating to the parchment quality, most directly, we can link it to the exposure to lime during the dehairing process. And this is often an indicator of how well the parchment maker was actually producing its parchment. So a controlled exposure to lime leaves less damage and produces therefore what we deem to be a higher quality parchment. This often not necessarily correlates with what the parchment looks like. You may have what you think looks like a very pristine white piece of parchment, but this could be due to a very long exposure to lime, and therefore the structural integrity of that parchment is actually not very good. So this is something we can actually measure. Aside from these intrinsic proteins, so mainly based on collagen, we can also look at other different kinds of proteins that have been used either as surface treatments to prepare the parchment for use, or those that have accumulated in the form of stains throughout its history. This requires a form of protein analysis that is a little bit more detailed and in depth. It's called proteomic analysis, but it does reveal a lot more information. Now, when comparing these two techniques, the benefits of protein analysis in general is that it's usually much cheaper and a lot quicker than DNA analysis. However, as you can see, you don't get all of the same information. So these are two techniques that we use in a complementary manner, and often perhaps use protein analysis as our initial pilot or our initial analyses to get a base level of what the species is and what may look interesting. And then those that actually warrant further analysis will then be passed on to DNA. So it's kind of a stepped process, and we would always recommend that people who are looking to do biocodicological analyses start off with a basic protein analysis to just get a feel of what's the quality of the parchment and what it's made of. The reason we've been so successful in using biocodicological techniques is because we use a non-invasive form of sampling. All of the work that I'm going to present later on would just not have been possible had we had to take destructive samples, because as we know, this is almost impossible to obtain. We do not want to actively be harming manuscripts and old books. The technique we used was developed together alongside with conservators in the Borthwick Archive in York. And it basically just requires the use of a standard PVC eraser that are approved by conservators and already used by them. The method is based on what we call the triboelectric effect. And it involves basically just gently wiping a piece of eraser very gently across the parchment. And through the electrostatic effect, just like you can see here with the sheep and the balloon, we're able to recover biomolecules, so both proteins and DNA, from the surface. And they effectively get entrapped in the tiny fragments of eraser that are created when we're wiping. So those bits that you normally, if you use an eraser, you wipe the surface and you generate these little crumbs that you throw away. Those little crumbs are the bits that we keep, and they're the bits that we analyze. We can extract all our biomolecules from there. If you're interested in seeing a more detailed explanation of the whole process based on the protein analysis, you can find a short video in this link, or by Googling YouTube parchment zooms. It's this first one that comes up. Once we have our biomolecules extracted, what can they actually tell us about the manuscript and its history? I've hinted about this beforehand when we were talking about the DNA and the proteins, but there are four main elements that we can explore and that can give us information about these manuscripts. The first is the species selection. So this can refer to economic practices depending on geographic distribution of animals, or preferences, or even scribal preferences for a particular animal that produces a particular type of parchment. We can also get information about the sex of the animals. So this can start to give us indications of slaughter patterns and if there is a preference of male or female animals to be slaughtered at certain times or for certain uses. We can find out if the manuscript was actively used for certain processes. So by looking at the surface treatments of these manuscripts, or more importantly, the stains that have been left behind by use, we can start to identify what kind of proteins appear in these stains. So can we actually have direct evidence of use of these manuscripts? And finally, we can also look at the environment in which these books or these manuscripts have been stored. So this is exploring the metagenome, or the microbiome, so those microorganisms that live on these documents. And this also ties in with conservation practices and maybe perhaps how we should be looking after these books, and if it should give us some more reasons to consider how we should be dealing with them. Our first example of species selection is the work we published a few years ago on 13th century pocket Bibles. So these were the small versions, the first complete Pandex, but in small format, produced in the 13th century in high numbers, and famed for their incredibly fine parchment. The folios are tissue paper thin. They're really incredible. There was a long-standing debate about whether there were specific animals used to make this incredibly fine parchment. Some people had suggested perhaps rabbit or even squirrel parchment, because it's so thin and fine. They assumed it would probably be made from smaller animals. Or there was also the option of using uterine or stillborn animals, which again would have very, very thin skin. So we sampled around 80 of these Bibles. In total, we sampled about 220 folios. And what we discovered is there's no rabbit, there's no squirrel, but we do find the three major forms of parchment, so calf, sheep, and goat. And what we find is the use of these different animals depends highly on their geographic distribution. So those countries that tend to use calf for their books would predominantly use calf for their pocket Bibles. Or in the case of Italy, where goat is very common, this was used also in their pocket Bibles. England has been more of a mixed picture. We found a little bit of everything. But effectively what we found was the making of this incredibly fine parchment is a technological process. They were working with the skins that were available to them and then just working them down to create this incredibly fine parchment. In addition to that, we also tested to see whether we could distinguish whether these animals were stillborn or uterine animals or older animals, and again just worked to that fine finish. Now, to do this, we did have to take destructive samples. So we used a modern comparison. We used a modern stillborn calf skin, and we have a piece of historic 18th century parchment that was given to us for a destructive analysis. And we did the comparison of these two. And by doing this special form of proteomic analysis, we were able to detect differences in proteins. Very specifically, the ones of interest were the levels of collagen 3 and fetal hemoglobin. And these two proteins we know are present in higher proportions in stillborn or intrautero animals and decline as once the animal is born and as it grows up. And we were able to detect the differences. So we were able to see this higher level of these proteins in our uterine calf versus the historic document. So potentially there would be a way of actually identifying whether these animals used were in uterine or not. However, we're still not at a place where we can do that non-destructively, but the potential is there. Another question that people often ask us or approach us with are supposedly exotic animals or parchments made from exotic animals, and can we confirm this? So we were approached by the National Leather Collection, who had an 18th century Ethiopian Bible that came in a satchel that was considered to be gazelle skin. So again, we took our rubbings, we had a look, and it actually turned out to be sheep. And this is often the case in all of the cases people have asked us to look at potentially exotic animals. It really always comes down to much more mundane or routine parchment animals, so sheep, goat, calf. We have yet to find the elusive gazelle or camel, but we're not losing hope. Maybe one day. But at the moment, it's interesting to see that what is assumed to be an exotic animal, maybe because just of the geography of where it was produced, is not necessarily the case, and they're actually using more standard animals for these manuscripts. Oh, just to warn you, there are a couple of slides where you can see here that I've put a no tweeting sign. This is just because they're still work in progress. They're still pre-publication. So please don't tweet these, but other than that, it's fine. So one of the aspects we talked about was the economic reasons for species selection. And a project that we've been working on for a few years now is that of the Aldine Press. So the Aldine Press was established in late 15th century in Venice by Aldus Manutius. And we've been looking at a lot of his early printed books that were actually printed on parchment rather than paper. So what you can see here is a timeline of the history of the press. And what's particularly interesting is in the time that Aldus was in charge of the printing press, the books he produced on parchment were made of goat parchment, which is not unusual for Italy, so this is quite expected. And it's all of quite high quality, shall we say, production of parchment. But what gets interesting is once the brother-in-law takes over, there seems to be, he maintains this goat for a time, but then shifts to calf. And finally, when the sons of Aldus take over the press, they shift completely away from the goat parchment and only use calf parchment. And we also start to see that they're creeping in are a few low quality versions of this calf parchment. So just by looking at the species and the quality of this parchment, we're able to actually reflect the different levels of, or the different times of ownership of the press, and how this has maybe influenced their choice, perhaps due to economic restraints, perhaps due to availability, we don't know. But it's starting to show us patterns that otherwise we wouldn't have realized it there. Another aspect that we've been interested in looking at is the complete analysis of one codex and what this can tell us about availability of resources. So here we have the example of a glossed Gospel of St. Luke produced in the 12th century in Canterbury. And it's held in a private collection, and we were given access to analyze every single bifolio from this book, and that's what you can see here in this collation pattern. And this book turned out to have a really interesting animal history. As you can see, it's made up of 3 animals in the parchment. There's calf, sheep, and goat parchment. And in addition to that, the bindings, which are contemporary, so they are 12th century, we found were of 2 types of deer. So the actual binding is roe deer, and the strap that holds the book together is either a fallow or a red deer. And you can see not only are they using in the parchment these 3 animals, but they're using it in a very specific manner. So they are interlacing calf with sheep in the first half of the book. Those choirs are all following a very similar structure of calf, sheep, calf, sheep. There comes a point where they start to use all calf. Then they start to introduce only 1 sheep. Then there is some goat, which we have no explanation for, but it's there. There's half a goat. It goes back then to original format. And then finally, the last few choirs are all sheep. Now, we don't know why this is the case. There are many possibilities. It could just be availability, and they are using what they have at that moment, and there could be a shortage of calf at the end, and they're just using sheep. It could be deliberate, because analysis of this book has shown that at this stage, in this final choir that's got the calf and the sheep, there is a change of scribe, and the main scribe, who has been doing the majority of the book, who is a very good scribe, is replaced by probably either a student or someone who's clearly not as good. Their scribal hand is not as good as the first one, and it could be that they were given this choir that started with the calf and the sheep, not considered good enough to have the lovely calf parchment, and were given just sheep, because obviously sheep is considered an inferior quality of parchment. So it springs up more questions perhaps than we'd initially thought. But just by looking at these very simple elements, it can reveal a whole past of the actual manufacture and use of these manuscripts that ordinarily you might not have thought of. Here we were looking at production treatment. So we were looking more specifically at purple dye. So different collaborators of ours have asked us to analyze parchment that has been stained with purple, or in the case of the Vienna Genesis, or blue dye in the case of the Blue Quran. And what we were able to determine was, in all these cases of purple dye, the parchment that was used was actually sheep parchment. Now, this is interesting because it has to do with the production or the actual method of dyeing the parchment. Now, I've put in this graph here because these are samples of parchments that have been analysed for lipid content, so for fat content. And what you can see is that sheep parchment has a lot more fat content than either calf or goat. And this is important because if you are dyeing the parchment, to have lipids or these fats means it's going to retain the dye in a much more efficient way than if it doesn't. So it could be a deliberate choice to be using sheep parchment for dyeing in these colours because of that fatty nature of the sheep. It's going to acquire that dye and keep the dye in a much more efficient way than if you used any other animal. Moving on to the sex of the animal, this is an example that we published a few years ago as well on the York Gospels, which we were very kindly given access to. Again, we did the protein analysis of the entire book, and you can see that the majority is of calf parchment, bar one folio of sheep. But what was interesting is we then took a subsection and selected 8 folios to do a DNA analysis on. And what you can see here are the results that we got. So from the 8 that we analysed, 2 unfortunately didn't work. But of the others, you can see that we found 3 parchments that were definitely female, only 1 that was male, 1 that we could say was calf, but we couldn't distinguish, and the final one, which was only human. Now, this is because this particular folio was one that had been heavily conserved in the early 20th century, and of course they've plastered over a silk gauze over the complete folio. So what we're picking up there is basically the conservators who have handled it since. But what is interesting to note is that we seem to have a majority of female cows versus male calves, albeit in a very small selection. But it does start to ask questions about what these slaughter patterns mean, because if we were focusing on a dairying economy, we would expect a lot more slaughter perhaps of young males, and that could then be used, for example, for parchment. But that doesn't seem to be reflected here. So although obviously it requires a much greater or larger data set, it does start to give us ideas about how we can use these techniques to interrogate manuscripts to actually give us information about slaughter patterns and the economies of the animals that are being used. Again, this is a pre-published work, so please no tweeting, but this is looking at the stains on parchment. So we've been working with the Wellcome collection and looking at a medieval birth girdle that seems to be quite heavily used and stained. And they asked us whether we could actually detect what these stains were and if that could provide any evidence as to how this object had been used. And when we did our proteomic analysis, we did find we could identify a lot of non-human proteins on those stains. So we found evidence for things that you can see here, such as honey, milk, egg, various cereals, including oats and wheat, peas, broad beans, and also I have added there a mouse, because we were also able to detect mouse urine, which we assume is due to the subsequent storage over the years, and at some point mice have probably got onto it. But it is a really clear example of how, using these biocodicological techniques, we can give direct evidence of use, of contemporary use of these artifacts, or in this case, the birth girdle. And it really reflects how we can help the interpretation of what has up until now been only done based on text and how people thought they might be used. But here we can see a direct evidence of them actually using it. So this is a really exciting field that we hope to explore more. The last example is the environment. So when we were talking about the microbiome, so these microorganisms that live on the parchment, this is the example again of the York Gospels, and on those same 8 samples that we did the sex ID for, we were also able to look at the microbiome. So the first thing you can see here on the left is the graph. We were basically comparing the microorganisms we found to standard microbiomes from Human Microbiome Project. So we compared it to the mouth microbiome, there's the stool microbiome, and then there's the nose and skin. And our samples plotted definitively with the nose and skin. So this is great because this is what we expected. The microbiome is to do with the handling of this object, and that's how our samples are plotting. On the right, you can see here, these are basically the different families of microorganisms that we're finding. And what's interesting is just to look at the patterns, and you can see here I've separated out in block colours what they refer to. So the first two columns are the original, are the additions of the York Gospel. So they were added in the 15th, 16th century, I think, they look quite similar amongst themselves, but a little bit different to what was the original 11th century Gospels, which you can see here in blue. And all those folios look very similar amongst themselves, but slightly different to the addition. The one in red is this heavily conserved page, which I talked about previously. And you can see it looks completely different. And that is to do with the microbiome of the people touching it rather than the environment over the years. And finally, in green, these are just some comparative documents. So these are legal documents held in the Borthwick Archive. And you can see that these all look different amongst themselves and from the York Gospels. But it is really interesting, by doing just this broad brushstroke interpretation of the microorganisms, you can see these same patterns of the original text, the later editions, and the conservation. And this just gives an example of the kind of things we could start to look into and can inform us about the history and possible conservation needed on these manuscripts. And the last thing I just wanted to touch on briefly was how can biocodicology help in digital representations? So ideally, all biocodicological information that we produce would be included in catalogue entries. That is what we would love to do. For every sample that we get a positive result, we would love to see this included. Of course, this does require a lot of effort and time, we know this, but ideally this could very much help people who are using exclusively digital resources. It gives them another layer of information. There would have to be some form of standardization, obviously, of the information that we provide for species ID. And we would like to include it in things like, much the same way that catalogue entries have collations. We would very much like to include a collation diagram like the ones that are produced in VisCol, which I'm sure a lot of you have used. And by including our species ID in the colour effects of VisCol, you could automatically see how a quire is composed. And if the protein analysis has been done, you could immediately see what animals are used on what folios. And we think this could be a very quick and a very visual way of interpretation. It's obviously always important to link the raw data. So all our raw data that is produced would, we have to do, every publication we do, we have to deposit our data anyway. So this is an obligation of the journals. So all our data that we publish is always deposited, and it has its own DOI. So it should be very easy to link back to these catalogue entries. So if people want to go back to the original raw data, they can. And then in a way, what we want to add is this level of sort of physical evidence for those people who are not able, especially now with all the constraints we have to access to libraries, and also just sometimes because things are very far away and they're not that accessible. This can help to add that layer of physical evidence that perhaps you don't always get with a scanned or digitized copy. It helps to remind people that these books are objects. They are a biological entity in themselves. They have come from animals, they have microorganisms still living on them. So it's a way of adding that layer of physicality that perhaps you lose when you're looking at a digital representation, but it's a way of at least being able to understand some of those biological properties and having that maybe help your interpretation of the text that you're looking at. And finally, I would just like to say thank you. We have a huge collaborative network, and obviously we wouldn't be able to do all of this work without them. So just thank you to all of our collaborators and libraries and archives who have generously sent us samples to continue with this project. Thank you.